Today we're gonna be doing an overview over vaccines. I'm Isaac, here with Yezer, Rima, and Bethany.
To first introduce our topic, we would like to show what a vaccine is and what the importance of vaccination is. A vaccine is a copy of a pathogen that stimulates the response in our immune system that will later create immune memory. All of this will be later discussed in further slides. It is important for the population to be vaccinated because this immune response allows us to once upon exposure to future infections, our body knows how to treat these infections. In today's presentation, we'll be talking about topics like the history of vaccines, new innovations like edible vaccines, and lastly, some sorts of types of vaccines.
First, we're gonna briefly go over the history of vaccines. They were first introduced in China in the year 1000 when someone grounded up scabs of smallpox, and then we get the first vaccination in America in the 1800s when Benjamin Waterhouse vaccinated his children for smallpox. Later, we have toxoid vaccine technology, which it uses the toxins of a vaccine toxins of a bacteria as a vaccine. And then we later have things like the polio vaccine, and then in December 2020 we have the COVID-19 vaccine.
The type of vaccines we're going to be talking about is live vaccines, killed or inactivated vaccines, toxoid vaccines, and subunit and conjugate vaccines. Live vaccines are weakened vaccines that are put into the body. Killed vaccines are vaccines which contains killed pathogens to produce a response from the immune system. Toxoids are inactivated toxins that build immunity, and then subunits are parts of the pathogens used for vaccines.
Our very first type of vaccine mentioned was live antigenated vaccines. These vaccines are made through a weakened version of the virus or a pathogen. It does not have to be specifically a virus. These weakened versions will still provoke our immune response, but don't actually cause too much harm or a substantial amount of harm in our bodies. These are made through passing the pathogen through cells or animal embryos to remove the ability from the pathogen to replicate in our human body and causes these illness. A strength of this vaccine is that they are very strong and they don't necessarily require booster shots. A weakness of this vaccine is that since they are alive, they do have the possibility of mutation as the pathogen is alive in our body. An example of this type of vaccine is a measles vaccine.
Killed or inactivated vaccines they contain pathogens that have been killed or inactivated but remain intact enough for the immune system to be recognized. It's made by the pathogen is treated to destroy its ability to replicate while preserving its structure. Some strengths of this vaccine is that there's no risk of disease or mutation, but a weakness of it is that it provides a shorter immunity compared to live vaccines. It requires booster doses and long-term protection. An example would be a seasonal influenza vaccine. Oh.
Our third type of vaccine is toxoid vaccines. These are vaccines that contain inactivated toxins from bacterium. What these what these, I don't know if I'm in frame or not. These bacteria, this vaccine includes this very specific toxin instead of the entire pathogen like the bacteria itself. These are made through extracting these are made through extracting the toxin from the bacterium. This toxin is neutralized using chemicals like formaldehyde or heat to remove its ability to actually cause an illness in the body but still be strong enough to create an immune response. A strength is that since it is specifically the toxin is what directly causes symptoms. So we are directly fighting against the symptoms. A weakness is that since it does require booster shots for longer immunity. An example of this type of vaccine is the tetanus vaccine.
A certificate vaccine is when the vaccine isolates a specific protein of to form a pathogen, and then for conjugate, it's vaccine produced by the pieces of coats from a bacteria. The strength is that for subunit, it's only specific components are used, and then for subunit, it's specific components are used, and then conjugates is that it's effective for people with weak immune system. The weakness is that they both would require multiple boosters. An example for subunit is hepatitis B vaccine, and then for conjugate, it's clonal vaccine.
This is some of the steps of vaccination. First, the patient would come in and take a seat. As you gather the materials, putting on your gloves, getting the correct needle, getting the right vaccination, and then when you're grabbing the correct vaccination, you would check the expiration date and the date of sale. Then you would prepare the medication for injection, and then after that you would select a spot and disinfect that spot to disinfect it. After that you would give the vaccine through most likely a fatty part in the arm, and then after the vaccine is administered, you would dispose of the needle properly and of the bottle vial. Then you would document the administration.
The immune system is when the immune system uses the body. The immune system is the body's defense mechanism against diseases, and it functions by identifying and attacking a pathogen that is affecting the body. And we can develop immunity by vaccines or by being exposed to the pathogens.
To further touch on what the immune system is, we have two different types of immunity in our body. The first type of immunity we'll be talking about is active immunity. Active immunity is passed down or developed through infection, either naturally or vaccination. This is when the body is exposed to a pathogen and the pathogen enters our system, and our immune system has a response, later creating immune memory. That will create immunity for further infections. Our second type of immunity is passive immunity. This is where a person transfers the antibodies to another, such as a mother breastfeeding their child. That's an example of passive immunity. Although they are both types of immunity, active creates creates or develops long-lasting immunity, while passive creates short-term immediate immunity.
Vaccines and the immune system. We have the vaccine is delivered, antigens presented in the cell. The T helper cell goes to the native B cell. Then the plasma B cell goes down to the vaccine's antigens. The antibodies secreted by plasma B cells to a specific antigen. Over to the other side, T helper cell goes down to the negative killer T cell, then to the active killer T cell, then its effector cell. Active T killer cell finds and destroys the affected cells. Immune memory is developed and into memory T helper cell, memory T cell, and then memory B cell.
Here is the vaccine and immune system, how they work. Formation of memory cell. After fighting the antigen, the immune system creates memory B and T cells. The memory cells remember the specific pathogen and help the body fight it for years. The benefits of the memory cells are that if they encounter the pathogen again, they'll keep fighting it. Vaccines are these vaccines are effective because they protect an individual for future infections.
Targeting viruses, bacteria, or both. Vaccines can target both viruses and bacteria by the approach varieties. The varying approach allows for vaccines to be applicable to various pathogens. The virus as a target. Vaccines for viruses include live, attenuated, inactivated, or subunit types. The function trains the immune system to recognize and attack viral particles. Bacteria as a target. Vaccines for bacteria include conjugate or toxoid types. Function prepares the immune system to fight bacterial toxins or components. All types of vaccines either target bacteria or viruses. The function protects infections by triggering the immune response specific to the pathogen. Bacterial meningitis vaccine. It is important for college students to get a vaccine because they live in much closer proximity to each other compared to regular people. It is also important for them to get a vaccine because the symptoms can come with containing bacterial meningitis, such as brain damage, hearing loss, and sometimes even death.
As we previously mentioned, certain types of vaccinations do have to have booster shots or additional vaccine doses. Additional vaccine doses can be recommended by a professional healthcare when they see that the sickness is relevant again, as in certain months, influenza is a lot more seen in healthcare, so doctors will recommend booster shots in those occurrences.
Now we're gonna talk about herd immunity. Herd immunity is when multiple people in a community receive a vaccination for something such as the flu or influenza. These people getting the vaccine help lower the risk of other people getting the vaccine because they are one less person that is able to contract the vaccine to pass it to someone else.
The laboratory process for live vaccine vaccines is that you have to first let the pathogen grow in a tissue culture, and then you have to, and then the pathogen will grow strains which will be filled in a syringe. Then this will cause the vaccine to spread across the host body. For subunit and conjugate vaccines, specific segments of the DNA, of the pathogen's DNA will be selected using tweezers, and then that would be added to yeast so it would be able to be read. Then that would be isolated by the yeast cell using a purifier, and then with that it would use a syringe to purify the antigen. For inactivated vaccine, to prevent pathogens replicating ability, use the tissue to grow new viruses, and then separate the copies, and then fill it with a syringe to kill the pathogen. For toxidoid vaccines, to create new vaccines, you have to grow the medium, and then you have to neutralize it to prevent harm to the host. And then when you're done, fill it with the syringe for the treated toxidoid.
Edible vaccines are a new innovative way of vaccination. Edible vaccines are made through introducing specific genes into either fruits, vegetables, or plants. These are then induced to manufacture and create these specific proteins that will help us, like other vaccines do. Some advantages of vaccine is that the need for needles and medical professionals is lowered. So that means we can have these vaccines. We don't have to go into the clinic. A disadvantage is that our bodies are more likely to build an intolerance, and the doses do come in inconsistent sizes.
You have to get flu shot every year because the virus is constantly changing and developing new methods to combat the vaccine. Two types of the vaccine. There are two types, which are A and B, and it has instructions for a new vaccine.
The career of an epidemiologist, basically what they do is they monitor what's going on around the world with new types of diseases and the mutations that occur in those diseases. They also help maintain the health of humans. Here in Texas, epidemiologists make about $67,000 per year. And then some knowledge that you would need is you need biostatistics, pathology, social science, and clinical medicine.
Vaccines are fundamental to safeguarding both individuals and communities by training the immune system to identify and fight against pathogens. Different types of vaccines are live, attuned, toxidoid, inactivated, and subunit, offer targeted protection against a wide range of diseases. Beyond protecting individuals, vaccines help prevent the transmission of diseases through herd immunity, thereby shielding vulnerable populations with continuous advancements such as the development of edible vaccines and flu shots. Vaccine technology remains a key factor in enhancing global health outcomes.
Here are references. Any questions? Oh, and also thank you for listening to our presentation.
Okay, I have some questions. We'll start with you, Zed. What is the difference between helper T cells and killer T cells?
Helper T cells are killer T cells are specific cells meant to kill infected cells. Killer T cells are a consequence of helper T cells stimulating naive killer T cells. So a T helper cell is just what activates these type of cells after antigen presenting cell has been signaled through the vaccine antigens. So what the cell does is that it captures these antigens from the vaccines and it presents it like a flag on its surface, and then from there the immune system sees it and like creates this response to it, and that's where T helper cells help out.
Alright, Rima.
Oh.
Could you explain what herd immunity is?
Oh, I'll come back to you on that.
Herd immunity. You want to get back to me on it?
I'll get back to you on that.
Alright, Bethany. How are restriction enzymes used in the creation of a vaccine?
You have to like cut DNA and make multiple copies to put the copies into like a host, so the host wouldn't develop, would get the disease again.
Isaac, what is the difference between conjugate and toxoid vaccines? Like the main difference?
Toxin vaccines use the toxin of the bacteria, and conjugate vaccines are they Can I get back to you on the last one?
Alright, good job.